Okay, so we're going to go ahead and get started. So thank you and welcome to this I3 session, an insightful webinar focused on today, non-invasive preimplantation genetic testing. So this event will cover the latest advancements in niPGT, including the current status, best practices for cell-free DNA analysis in the IVF laboratories, and practical experiences in PGT, tubing, and genetic sampling of the blastocoel. This webinar today is kindly sponsored by Thermo Fisher Scientific. We'll keep the bios short, but the full extensive resume of the careers of the experts that we have today can be found on the I3 website. I'm joined by two co-hosts today. The first, Dr. Berta Younis. She's an experienced embryologist and currently serves as the Europe, Middle East, and Asia Market Development Manager for Reproductive Health for Thermo Fisher Scientific. Berta drives the EMEA marketing strategy and supports visionary life science organizations in expanding their lab genetic testing capabilities, and I am so happy to have you here.
It's my pleasure to have you also, Catherine. So let's introduce you to our audience. Catherine brings with her over 20 years of experience as a clinical embryologist, collaborating with leading IVF centers throughout the United States. So throughout her career, she has focused on enhancing fertility outcomes and expanding patient access to care. Catherine also consults on diverse aspects of clinical operation, specializing in laboratory design and management. She also leads Fertility Tomorrow, an embryology group, and we are very happy to have you here, Catherine.
Thank you, Berta.
I will be also joined, you're welcome. I will be also joined by Professor Jan Traeger-Synodinos, who is a professor of genetics with a Doctor of Philosophy from the University of Oxford. A proven track record of working with human medical genetics in public healthcare, applied research, and higher education. And I am sure the Greek audience that we have here will recognize her very well. She specialized in molecular genetics of rare diseases, pre-implantation genetic testing, and recognized nationally and internationally for contributing in the field of human medical genetics, especially with the hemoglobin disorder and pre-implantation genetic testing. It's a pleasure to have you as well, Jen.
Thank you very much, Berta. Thank you very much, Catherine, as well. And it's a pleasure for us to have everybody here with us as well, with what promises to be an extremely exciting and enlightening webinar. I'd like to remind everybody when they want to ask their questions, if they can put it in the Q&A section rather than in the Zoom chat, so that we can gather them and select some of them for the question and answer sessions, especially at the end of the whole seminar webinar tonight. I'd like to introduce our first speaker, Dr. Gary Nakhuda. He's going to talk to us about the current status of non-invasive preimplantation genetic testing for aneuploidy. He's an associate clinical professor at the University of British Columbia in Vancouver. Before moving to British Columbia in 2011, he was at Columbia University, where he completed his residency, his fellowship, and he served on faculty for 5 years. He's the founder and co-director of the Olive Fertility Center in Vancouver, and the lead author on the recently published paper and results of the NICEST trial, which was recognized as a seminal contribution in fertility and sterility.
Thank you. Thanks again for the kind introduction. I'm so honored to be invited to speak to this esteemed group from all over the world, and I'm a little bit nervous to speak to this large group. I'm just a simple clinician, but I'll be reviewing the current status of niPGT-A, mostly from our clinical perspective, and presenting data from our recent non-selection trial. So just my first conflicts of interest: I'm a co-manager at Sequence 46 and a shareholder in the Fertility Partners. This is the disclaimer that the Thermo Fisher lawyers asked us to display, and I'll get into it. So PGT-A is still controversial for many reasons, but one of the major concerns is the possible harm from the biopsy posed by PGT-A. And this is a divisive issue. There's two camps. Some would say yes, the biopsy is in fact harmless. And there have been at least two prospective studies, both from the RMA group, the most recent published in 2021 or so. And it did not demonstrate a difference in the sustained implantation rates between biopsied and non-biopsied blastocysts. On the other hand, there have been several studies, both prospective and retrospective. This is a large one of the review of the SART database of nearly 32,000 cases, which suggested that PGT-A reduced the live birth rates cumulatively, citing the effects of the biopsy as a potential source of the inferior outcomes. And while this may still be a sensitive point of contention, and there's most certainly a range of outcomes dependent on the skill of the embryologist or the lab, I think any logical person can agree that there is at least the potential for risk, given how much skill is required for this delicate procedure. So if asked the question, why are we interested in non-invasive, I would posit that given the choice between an invasive or non-invasive anything, most of us would choose the latter. Specifically for the context of this discussion, non-invasive PGT-A potentially offers to eliminate the risk of the biopsy. It could require less skill from the embryologist, which potentially increases access. And some have said it may lower costs, not so sure about that yet. Maybe it's less labor intensive on the embryology side, but it remains to be seen what happens on the sequencing side. Recently, the ASRM Practice Committee just put out this their statement about routine PGT-A, and it suggests that the routine use of blastocyst biopsy with aneuploidy testing in all infertile patients undergoing IVF cannot be recommended. So this also supports that the innovation of new technologies such as non-invasive PGT-A may help offer better outcomes, hopefully in the future. So there are several approaches that could be considered non-invasive screening tools, including morphology, which we do every day, and time-lapse imaging, which is gaining traction. And this study here was used a mass spec for metabolomics from the blastocoel fluid, which is, I think, one of the first published methods as a less invasive analytical method, although one could argue that obtaining blastocoel fluid is still not truly invasive. However, the attention has shifted towards cell-free DNA in the spent culture media as the most promising alternative to conventional PGT-A. The first reports documenting the possibility of using cell-free DNA in spent culture media were published in 2016. Musa Shamonki's group used array CGH on trophectoderm and spent culture media on 57 blastocysts. They found detectable DNA in all spent culture media samples eventually, but only 2 passed quality control, but both showed the same copy number in the trophectoderm biopsy and the spent culture media samples. So this was obviously very low efficiency, but it served as a proof of concept that niPGT-A was possible. Several subsequent studies were published in the next few years attempting to validate niPGT-A. The one that I would say was the most impactful was this one authored by Dr. Rakowski and published in the Proceedings of the National Academy of Sciences in 2019. And they had donated embryos that were previously biopsied for PGT-A for clinical purposes. They were thawed, and the spent culture media was collected after 24 hours in culture. Then the trophectoderm biopsy and the spent culture media results were compared to the analysis of the whole embryo by NGS. And they reported superior concordance of the spent culture media to the whole embryo relative to the compared to the trophectoderm biopsy. And they concluded that non-invasive PGT-A has the potential to be superior to trophectoderm biopsy for PGT-A, while avoiding the trauma associated with TE biopsy. So it seems like every month there is another study that's published seeking to validate niPGT-A by correlating ploidy or chromosome concordance between the spent culture media and the TE or the whole biopsy. And different pre-analytic and analytic techniques have been used to varying degrees of success. This is the most recent study I came across, which is in the September FNS from the IVF Boston collaboration, which used donated day 5 and day 6 blastocysts. They thawed them and they cultured for an additional 8 or 24 hours to determine the optimal timing that allowed successful analysis of the spent culture media. And they reported greater than 90% concordance with the whole embryo analysis. So it seems like we're getting better. And although we're improving, the common challenges encountered by all investigators include maternal contamination, amplification failure, non-interpretable results, and the correlation studies really don't give us a picture of the true clinical value. Furthermore, we're still not clear where the cell-free DNA is actually coming from in the spent culture media. If you rule out exogenous contamination and maternal contamination, the thought is that this DNA is being released via apoptosis. It may be coming from either the trophectoderm or the ICM or both, and it appears to happen in both euploid and aneuploid cells. So all these studies have helped to establish the foundation for niPGT-A, but there are still significant limitations to the body of literature. For example, many of the studies use methods that are not consistent with routine embryology practices, thus may not be really clinically practical. Also, it's important to highlight that benchmarking to trophectoderm biopsy assumes that the trophectoderm biopsy is accurate and deterministic, when we all know that that, in fact, is not always the case. Furthermore, in studies where the embryos have been selected by niPGT-A, but they were also biopsied, really negates the advantage of a truly non-invasive procedure because they were biopsied. And also, perhaps most importantly, if only putatively euploid embryos are transferred, we are never really scrutinizing the false positive rate. So an unbiased study must transfer embryos blinded to the analytical results. So what is a non-selection study? This is what we needed for a non-invasive PGT-A. And even though the term has crept our way into the vernacular, critics would argue that this is not a standard epidemiological term, but it refers to a blinded prospective observational cohort design, where the embryos are selected by routine criteria, but the analysis of the archive test sample is only released after the clinical results are known. So this allows us to calculate the clinically relevant predictive values by comparing the actual outcome to the outcome that would have been predicted if the test sample had been used for embryo selection. The goal is to ensure that the error rate is as close to possible, ideally less than 5%, to avoid harm to patients, and ensure that competent embryos are not being routinely discarded. We can attribute the term non-selection to the prolific RMA group. Their most recent non-selection study investigated the positive predictive value of whole chromosome aneuploidy detected with conventional PGT-A, and they stated that the primary goal was to predict the failure to deliver. And they reported a positive predictive value of 100% for whole chromosome aneuploidies. However, they also distinguished abnormal copy number variants other than whole chromosome aneuploidy that they coined as uncertain reproductive potential. And these, in fact, showed poor positive predictive value with sustained implantation rates of as high as 41%. So it's important just to define what predictive value means for aneuploidy screening. So a true positive is an abnormal NGS profile associated with a non-viable outcome, so failed implantation, spontaneous miscarriage, or clinical aneuploidy. And a false positive is an abnormal result associated with a viable outcome. For an abnormal copy number, the positive predictive value is what we're interested in. A true negative is a normal NGS profile associated with a viable outcome. A false negative is a normal NGS profile associated with a non-viable outcome. And for a normal copy number, the negative predictive value is what we are interested in. Positive predictive value and negative predictive value always depend on the prevalence of the condition in the population, or you can consider that the pretest probability. In a low prevalence population, where the condition is relatively rare, there's actually a higher risk of false positives. Thus, a low prevalence population is a greater challenge to the positive predictive value. And predictive values are more relevant than sensitivity and specificity when people are being screened, whereas sensitivity and specificity are the best indices for analytic or pre-clinical studies. But we're focusing here on the predictive values. So we endeavored to emulate the so-called non-selection study design to investigate the predictive values of non-invasive PGT-A. Our results were published in FNS in July of this year, and we were honored that it was deemed a seminal contribution. My co-authors are the excellent team at Sequence 46, Sally Rodriguez, Sophia Tormasi, and the very talented Catherine Welch, who happens to be on this call. The trial was nicknamed the NICESS trial, which stands for the Non-Invasive Chromosomal Screening for Embryo Selection trial. So the objectives, the primary objective was to investigate the positive predictive value of whole chromosome aneuploidy. In other words, does aneuploidy detected by niPGT-A accurately predict non-viability? We would have to transfer the embryos blinded to the results, and this would reveal the false positive rate. The predictive values of abnormal copy numbers other than whole chromosomes were also of interest. So we would characterize those as well. The secondary objectives were to investigate the QC parameters associated with the NGS profiles with niPGT-A, and just to add a larger descriptive data set to the literature, although these embryos were not connected to a clinical outcome. So we have quite a bit of data in our paper that I invite you all to read, but I'll stick to the primary results for the rest of this lecture. The inclusion criteria were patients under 35 at the time of retrieval who had an indication for ICSI and a plan for a deferred frozen embryo transfer of a single embryo. Patients were excluded, if they had recurrent implantation failure, recurrent pregnancy loss, required a trifecta biopsy, or had a plan to transfer more than one embryo. So why do we focus on the young patients? So again, given the lower prevalence, given the lower prevalence of aneuploidy relative to the older age groups, with a relatively higher risk of false positive, we could expect the younger patients to be the best challenge for positive predictive value. This was intended as a pilot study, so sample size was not determined by a power calculation, but we targeted a sample size of 120 patients based on an expectation of approximately a 50 to 55% live birth rate per single embryo transfer in this group. And we estimated that if the false positive rate was 10%, we should have seen about 6 to 7 sustained implantations that would have been incorrectly classified as non-viable. So essentially 6 to 7 false positives were expected. Our clinical methods were relatively straightforward. In the laboratory, performed a culture media change on day 3, at which point the embryos were moved to individual 30 microliter drops. Now, that's a relatively large drop size compared to other studies, but we wanted to keep it as consistent with standard embryology practices as possible. The blastocysts were vitrified. No hatching was performed before vitrification. Spent culture media was collected in PCR tubes, de-identified and shipped to the reference lab. When the patients returned, the embryos were selected based on standard grading criteria, and the results were unmasked after the clinical results manifested. So after a failed implantation, a miscarriage, or after 13 weeks of a viable pregnancy. And standard prenatal testing was offered to all patients. We used a validated PGS platform. Apparently, I'm not supposed to say the name, but it's all in the paper. The technique involved the whole genome amplification with 150 to 2,000 reads per sample. And the QC metric of the median absolute pairwise difference less than 3 was the threshold to pass QC. The primary NGS interpretations were either failed QC if the MAPD was above 0.3, no copy number variant detected if the copy number fell between 1.5 to 2.5 for all chromosomes, or an abnormal copy number variant if it was less than 1.5 or greater than 2.5 for any chromosome. We then also stratified the abnormal copy number variants to subcategories. The primary interest was whole chromosome, which we called if it was 3 or less whole chromosome aneuploidies. And then uncertain reproductive potential, or ERP, would be other abnormalities like segmentals, combined whole and segmental, or chaotic profiles, which were more than 3 chromosomal abnormalities. We also stratified the copy number variants, the abnormal copy number variants, to either uniform, if there was either less than 1.2 or greater than 2.8 copies, or intermediate, which we would, synonymous with mosaic for trophectoderm biopsy, if the copy number was between 1.2 to 1.5 or between 2.5 to 2.8. The sex chromosome interpretations were a little tricky. We did see all the typical sex chromosome aneuploidies, but we did see some patterns that were not typically seen with conventional PGT-A, like non-integer ratios of X to Y. So when this occurred, we classified according to the autosome number, and then we called it sex undetermined. We analyzed all the typical clinical outcomes, but again, the primary outcome of interest was non-viability, which included implantation failure, miscarriage, or a clinical aneuploidy. Now, live birth rate obviously is usually the primary outcome of interest in our field, but because positive predictive value of abnormal results was being scrutinized, we were actually more interested in non-viability. And to the results, so of 120 single embryo transfers, when unmasked, these were the niPGT-A results. 9 or 7.5% failed QC. In 75 or 62.5% of the total, there was no CNV detected. And an abnormal CNV was detected in 30% or 36. Of these, 18 were whole chromosome aneuploidies, and the remaining 18 were a combination of the uncertain reproductive profiles. And then of the whole chromosome aneuploidies, 5 of them were of the intermediate copy number extent, and the 13 were uniform copy number variations. So if we look at the clinical outcomes, stratified by NGS interpretation, overall 64 of the 120, or 53.3%, resulted in a sustained implantation. We actually followed these to live birth, but for the paper they're labeled sustained implantation. For the primary categories, for failed QCs, 6 out of 9 actually were sustained, so 66.7%. When no copy number was detected, 57.3% were sustained. And collectively for all of the abnormal copy number variants, the sustained implantation rate was 41%, which is still pretty high. However, when stratified, if we looked at the whole chromosome aneuploidies, there was only 1 sustained implantation out of 18. The other uncertain reproductive potentials had a very good sustained implantation rate, 14 out of the 18 actually. So to the primary outcome of interest, this is where we calculated the positive predictive values and calculated the relative risk relative to embryos with no CNV detected. So again, in aggregate, if you look at all the abnormal copy number variants, the positive predictive value was 58%. The relative risk compared to no copy number variant was 1.37. This is not statistically significant. However, if we look at the whole chromosome aneuploidies, the PPV was 94%, and the relative risk was 2.21. This was highly significant. The others, again, were not significant. When the extent of the CNV was substratified, so for whole chromosome aneuploidies, we broke it down into either the uniform or the intermediate. If you look at the uniforms, there were 0 out of 13 that were sustained, or 100% positive predictive value. Out of the intermediates, which can be considered analogous with mosaics, there was 1 that was sustained, which gives it an 80% positive predictive value. There's actually no difference when we compared the positive predictive values statistically, but obviously there was 1 live birth. And this was a trisomy 20 when we unmasked the results. We were able to there's another interesting case of a patient who appeared to have a viable pregnancy, but when the niPGT-A result was unmasked, it revealed a trisomy 22, which we at first thought was a false positive, but the patient pursued amniocentesis for suspicious anatomic findings. And then the amniocentesis actually revealed a partial tetrasomy 22. So even while it wasn't the exact structural aneuploidy, niPGT-A did in fact flag the abnormal copy number of the correct chromosome. Now, the sex chromosome ratios were significantly skewed female in samples overall, and also in samples with no copy number variant detected. There was predominance of XXs, and the difference in proportions was significant. However, if you look at the any of the abnormal CNVs, we can combine those. There's actually no difference in the sex chromosome ratios, which is interesting. And then we were able to confirm the sex predicted by niPGT-A clinically to either birth outcomes, NIPT, or amnio. And 54 with informative sex chromosomes and clinical results were available for comparison. Overall, 74.1% were consistent. All 14 that were misdiagnosed were clinically XY, but called XX by niPGT-A. But conversely, when XY was identified by NGS, male sex was always confirmed clinically. We also then performed a post hoc analysis. The purpose of aneuploidy screening is to accurately identify non-viable embryos, but also to enrich the remaining pool to improve the probability of selecting a viable embryo. So if we deselected all of the whole chromosome aneuploidies, we would have been left with 102 transfers as opposed to 120. This would have resulted in a sustained implantation rate of about 62% compared to 53%. So about 9% higher, which we would say is pretty important, but this was not clinically significant. And if we deselected only the uniform whole chromosome aneuploidies, so kept the intermediates in the pool, the sustained implantation rate would have been 59%, nearly 60% compared to 53%. Again, a relatively important difference for our patients, but not clinically significant. Although we think this is probably just type 2 error, we needed a bigger sample size. So ours was the first blinded study to report the clinically verified prognostic accuracy of CNV abnormalities detected with non-invasive PGT-A. For the whole chromosome aneuploidies, we had a very good positive predictive value of 94.4%, which was statistically significant. And there was a high relative risk of 2.21 compared to embryos with no aneuploidy detected, no copy number variant detected. However, the other copy number variants of uncertain reproductive value had very poor positive predictive value. And if we had deselected these embryos in real life, we would have discarded a lot of viable embryos. So this just emphasizes what I think we all already know by this point: all abnormal NGS profiles are not alike. Just a word on the negative predictive value of normal niPGT-A results. A negative predictive value is always expected to be lower than positive predictive value because embryos may fail to implant for numerous reasons, undetectable by any single testing method. In our group, the negative predictive value, which is synonymous with sustained implantation rate, was 57%, which most of us would say was pretty good. And even though it's not a direct comparison, if you look at the STAR trial, the sustained implantation rate was 49%. And if we look at our own internal patients who have had conventional PGT-A, the sustained implantation is about 55%. So at the very least, with a non-invasive method, the negative predictive value was as good as conventional PGT-A. So the biggest challenge from our study was the ability to consistently obtain informative niPGT-A results. If we combine the undetermined or uncertain results, out of the 120 patients, there are a total of 27 between the failed QC and the uncertain reproductive potential. It's 22.5%. It's probably a little bit too high for clinical use. We definitely encountered maternal contamination. It's interesting that the sex ratios were equal in samples with abnormalities, but unequal in other cases. The 46XY and non-invasive PGT-A results always were clinically confirmed, so 100% accuracy when we see that. But the overrepresentation of 46XX alludes to maternal contamination. And this is something we need to be careful about, particularly as we get to older patients where there's a higher risk of aneuploidy. We might be masking those abnormal results. So next steps, we're planning another trial. We're going to focus on an older cohort. And again, the risk of false negatives will be higher. We suspect false positive rates should be even lower, but it's going to be crucial to have informative results. We've already have some plans to refine our techniques, both pre-analytically and analytically. And this is just a speculative question, but I've always been curious, and I've spoken to others in the field who have the same hypothesis that maybe abnormal copy number variants, different types of copy number variants are indicative of disparate outcomes. So in other words, is it possible that when you see a segmental aneuploidy in the spent culture media, is it actually a predictor of sustained implantation rather than of failure? So we don't have any kind of neat biological explanation for this, but it's interesting. So the goals of non-invasive PGT-A, in my opinion, are to obviate the need for trophector during biopsy for aneuploidy screening, and I'm willing to bet we will get there soon. This was summarized by Cooper and Viotti recently in reference to mosaic results in conventional PGT-A, but it rings true for non-invasive PGT-A too, and it's always been designed to be a screening test to reduce time to baby, not to improve cumulative live birth. We can't make embryos any better than they are, but we certainly shouldn't be reducing cumulative live birth. And PGT-A should be considered a ranking tool to help pick the embryo more likely to result in a healthy live birth and less likely to end in a bad outcome. But the binary classification of NGS results is simplistic and inaccurate. So I think we are gonna have to consider a multi-omics approach. I think non-invasive PGT-A with a high positive predictive value can help deprioritize the abnormal embryos with the whole chromosome aneuploidies, and we'll probably need another technique, which could be AI imaging, time lapse, what have you, to select the remaining embryos that have with a negative predictive value. So pick the best embryos. We're just going to have to use a couple different techniques. And I would like to just conclude by, again, thanking the amazing team at Sequence 46, Sophia, Sally, and Catherine, my incredible embryology team here at Olive. We couldn't have done the work without them. And thank you all for listening. I hope to collaborate with all of you. We are planning that second trial. So if anybody is interested in participating, please get in touch, and we will get details out as soon as we have them.
Dr. Nakhuda, thank you so much for that very detailed and informative presentation. I wanted to remind everybody on the call today to go ahead and put any questions you have in the Q&A and not in the chat, and we will collect those and save them for the end for the Q&A session. Right now, we have time for just a single question for you, Dr. Nakhuda. So I think that I'm going to go ahead and with one of my own, which is, we don't often get to talk to clinicians necessarily about how they will counsel and whatnot. So for you as a early adopter of this type of technology and a believer in niPGT, and how will you counsel your patients differently?
Well, we're not using it clinically yet, but I assume when we do, I will basically can reiterate that this is not a diagnostic test. This is a screening test. We're just trying to get to the best embryos and get the patients to the best outcomes as soon as possible. But I think, there will probably be times when we have a result that appears to suggest that it's a non-viable embryo, but a patient may wish to transfer it anyway. And that's just, something that we're going to have to discuss with them, on an individual basis as we do currently. with mosaic outcomes and even segmental outcomes with trophectoderm biopsy. I think we just can't be so rigid and hard-lined with these results.
Thank you again. We'll continue with more questions at the end of the session, but right now it's an honor for me to introduce Dr. Anil Biricik and his talk, Recommendations for the IVF Laboratory for the Most Effective Cell-Free DNA Analysis During Non-Invasive PGT. Dr. Anil Biricik received his PhD from Sapienza University of Rome in 2011 with his work on genetics of endometriosis. Dr. Biricik has published more than 30 peer-reviewed articles as author and co-author in molecular biology and reproductive genetics fields. He is a scientific director of the IVF and PGT business unit at Eurofin's Genoma Group, Italia, and his main research areas are the genetics of female and male infertility and novel application methods in preimplantation genetic testing. Welcome, Dr. Biricik.
Okay, Catherine, thank you very much again for your introduction. And my presentation will be some recommendations for the IVF laboratories for the effective cell-free DNA analysis during non-invasive PGT applications. This is my disclosure and disclaimer. The presence of cell-free DNA molecules in sperm culture media and blastocell fluid has been demonstrated more than a decade ago, and this opened a new possibilities for the genetic testing of preimplantation embryo without need to embryo biopsy. And the exact mechanism of cell-free DNA release into sperm culture media and blastocell cavity is not very well known, but apoptosis is most likely cause. And the recent study by Ana Domingo Miles et al. Showed that there are also some mechanical mechanisms during the blastocyst expansion may cause the release of these small DNA molecules in the sperm culture media. And non-invasive PGT applications can bring many advantages. First of all, non-invasiveness and easy accessibility for many IVF laboratories without needing any expertise, and probably potentially reducing the costs arising from the embryo blood biopsy equipments, and especially for the countries where there are some legal and ethical restrictions with embryo biopsy. And also it can be applied on the poor morphology embryos when embryo biopsy is not possible. However, there are also some limitations and restrictions for the potential use of non-invasive techniques, like first of all, contamination risks from various sources and standardization issues. And currently it can be also offered as a prioritization test, and especially it is much more adapted for aneuploidy screening and for PGT-A, and it is less effective for PGT-M or PGT-SR applications. And the detection of chromosomal mosaicism is also challenging. So if we look at the origin and the amount of cell-free DNA in sperm culture medium blastocell fluid, first of all, the one comprehensive study by Rodriguez et al. Showed that the quantity of cell-free DNA in sperm culture medium is expected about 3.2 until 12.6 picogram. It is more or less in medium if we say the 6 picogram is like a genomic quantity of a single cell, and this is more or less the same amount of cell-free DNA we expect in blastocell fluids. And if we look at the origin of these cell-free DNA molecules, that basically we expect them from trophectoderm or inner cell mass, and but not only, also of course from maternal cumulus cells, polar bodies, sperm cells, or even some exogenous DNA may also contribute to the presence of this cell-free DNA. And also we should consider the additional biomarkers like proteomic, metabolomic products or exosomes or microRNAs. Sampling methods in non-invasive PGT-A, basically there are two approaches, and the first is the blastosynthesis. I think next speaker will give more details on this, but briefly, in this technique, the sample collection needs an expertise approach by an ICSI needle entering the blastocell cavity and aspirating the blastocell fluid. And we are talking about like 0.3 or 2 nanoliter of quantity. And This method is a little bit controversial due to its minimal invasiveness. Span culture media collection instead is totally non-invasive, and since it doesn't need any embryo manipulation, and it is very simple for collection. But of course, we should consider all the presence of biomarkers together with cell-free DNA in the span culture media. And another approach is the combination of both blastocell fluid and the span culture media by zona drilling. In this method, the blastocell fluid can combine with the go out from the blastocell cavity and contribute to the span culture media. And the concordance studies are showing the highest concordance once span culture media and blastocell fluid combination applied, analyzed together. But this method also is being considered as minimally invasive. If you look at the workflow of non-invasive techniques, after the ICSI fertilization, embryo culture has been done until day 3 or day 4. And the 4th day of the embryo culture, medium droplet is being changed, and the culture is being prolonged until day 5 or day 6 until blastocyst level. And during this culture, and together with the embryo droplet, one empty droplet, medium droplet, is being can be used at the same conditions as a blank sample. And the blastocyst level embryo, while blastocyst embryo is being vitrified, span culture media can be collected or blastocell fluid and together with the medium blank, and these samples can be amplified and analyzed by the downstream applications like NGS for aneuploidy screening. In the invasive method, instead, as we know very well, at the blastocyst level, trophectoderm biopsy is being done while the embryo is being vitrified. These blastocyst samples can be analyzed by NGS, and the concordance studies are usually designed for the comparison of genetic results from these both samples, trophectoderm sample or cell-free sample from the same embryo. If you look at the NGS plots coming from the span culture media, they are quite clear and very similar to the trophectoderm sample, and we can easily determine the euploid X6 or XY samples, and also some deviations in NGS plot like trisomies or monosomies, as we can see the partial duplication of chromosome 9, or as we can see the bottom image, the monosome of chromosome 4, trisome of chromosome 21, and monosome of 22. It's quite easily determined. It can be determined easily. And for the validation of non-invasive PGT applications, most of the studies has been done by concordance analysis. And if you look at in the literature, of course, there are only a few of the publications available in the literature, but if we can see, there are many the different applications, different study designs, especially for the amplification methods or culture drop size are different, and some samples also using some studies showing the assisted hatching or frozen solved embryos, and as a consequence, ploidy concordance are changing from one study to another. And if we look at the blastocell fluid studies, usually amplification rate and the concordance rate is less when we compare to the span culture media. And all these data shows us that especially the standardization and the correct study design for non-invasive PGT applications are essential. So if you look at the challenges for an accurate cell-free DNA analysis, and first of all, DNA contamination is the first issue. And as we know, the main sources for the contamination is the maternal albumin cells or polar bodies. If we look at usually basically false negative results coming from the span culture media are female profile, which is demonstrating the presence of the maternal DNA. Or paternalogeny deriving from the sperm cells may also contribute the DNA contamination. Or it is very rare, but some exogenous sources from IVF laboratory can also cause DNA contamination. And another study is demonstrated by Hammond and colleagues is also manufacturing process also may have some traces of genomic DNA in commercial media. So another challenge was the technical the challenge for the optimization of sample volume and handling. Usually spent culture media volumes are reported are between 5 to 30 microliter, and as we know, these volumes could not always be compatible for the following target PCR or whole genomic amplification protocols, because we expect that if the initial sample volume is higher, then the amplification efficiency will be lower. And if we consider that the recommended initial reaction volume for the PCR or whole genomic amplification is 2 or 3 microliter, these volumes are will have an important impact on the amplification efficiency. And if you look at the blastocoel fluid, instead the volume is 0.3 or 2 nanoliters. These are very small quantity, and these volumes are quite compatible for PCR or may have created some problems with the handling. In fact, these the studies with blastocoel fluid are showing less performance for amplification. But these may come from the handling of the volumes or some biological factors, as Magli and colleagues showed that if the blastocoel fluid amplification is less, then this also showing higher pregnancy rate, or in a good amplification is showing the lower pregnancy rates, which shows the probably apoptotic status of these embryos, and they will have which have less implantation potential. And another challenge is the quantity and integrity of DNA, and the amount of cell-free DNA is, we are talking about 3 picogram, and different than the other cell-free DNA analysis methods like non-invasive prenatal testing, these small amount of DNA requires an effective full genomic amplification process. And fragment sizes of cell-free DNA are absorbed in 2 populations, like 160 and 220 base pair, and 300 and 400 base pair, as we see in this study. And these also should be considered as a challenging factor for a successful amplification. Or some other methods like some amplification methods like multiple displacement amplification requires a continuous genomic DNA template for the amplification, and this may also limit for amplification of cell-free DNA molecules. If the targeted amplification protocols will be used, then this may require some optimization of the small amplicon designs of PCR and NGS library preparations. So all these factors are showing us the adaptation of PGT and IVF laboratories for non-invasive PGT applications. The first important thing is to preventing the contamination. It is very basic, but some specific precautions should be taken during the IVF lab protocols, like using mask, surgical gown, hair cap, and DNA-free hood surfaces, and fresh adequate of culture media for individual pipettes or for sample aspiration, and so on. Oocyte denudation is very crucial to avoid maternal contamination, as we expect from granulosa cells or cumulus cells. ICSI is always preferred to avoid paternal contamination, and serial media washes during the culture of the culture droplet is very important. And in a recent study, also showed that instead of single droplet rinse, if the sequential rinse, so more than one droplet, is being applied, then the concordance rates are being higher. And this is also showing that the early stage of in vitro development maybe leaves much more apoptotic cells, and it is not very good representative of the embryo. So that's why the culture droplet change and the sequential watches are important. And from PGT lab, of course, we have to consider the contamination detection markers like SNP or STR markers, which can be the indicators for maternal or external contamination, and also blank sample, which is cultured together with the embryo, is important to check the external contamination control, and also data analysis methods like specific NGS bioinformatic pipelines for data analysis and interpretation can be also very helpful to understand eventual contamination. And the volume of spent culture media is an issue for the standard IVF procedure, because we know that in the IVF laboratories and embryologists, they don't want to reduce the culture droplet volume, but it is essential to find an optimized volume for an ideal DNA amplification after cell-free DNA collection. And according to the studies, the 5 or 10 microliter is ideal for effective amplification, but also 15 to 20 microliter is quite acceptable. And if a time-lapse system will be used, and then a culture dish with single wells and separate media drops should be used instead of multiple micro wells to avoid any cross-contamination between the embryos. And if the culture volumes optimized, then both sequential media or continuous single media would be adapted to non-invasive PGT protocols. And from the PGT lab, of course, the volume and the amplification protocols should be adapted to this culture media volume. So in our laboratory, for the initial pilot study, we did some amplification trials with different culture media droplets ranging from 5 to 30 microliter. And after whole genome amplification, DNA concentrations were higher in 5 microliter and 10 microliter initial culture droplets volumes. And it was less than 20 and 30, but even 20 microliter were giving almost a very good results for amplification. And quantity and integrity of cell-free DNA. This is also very important, because we should know that if we have enough concentration of cell-free DNA, then we expect very good amplification results. And the ideal culture time to collect cell-free DNA from spent culture media is between 24 or 48 hours. And preferably, the better results is coming from day 4 to day 6. For this aim, and our group has studied the concordance analysis study by using 482 spent culture media and the biopsies, the corresponding biopsy trophectoderm biopsy sample. And we observed that the informativity and the ploidy concordance results are better in day 4 to day 6 spent culture media samples. And the DNA concentration after amplification, as we expected, from day 3 to day 6 group was higher. But of course, this is one expected results, because more embryos, much more time remaining in spent culture media, more cell-free DNA is released. So that's why we set up our ideal embryo spent culture media collection time, like period like day 4 to day 6. So this is also another issue for the culture time, to prolong the culture time to the until day 6 or even day 7. But there are some studies are giving very promising results, even if the embryo culture is being prolonged until day 7. The aneuploidy results and the embryo implantation results in day 7 are quite similar to day 5 to day 6. So this should also be considered as prolonging culture time shouldn't have any negative impact on the embryo implantation. So another important factor is spent culture media integrity. Because we are talking about very small cell-free molecules, DNA molecules, so we are very sensitive for the degradation. So as soon as media sample has been collected, they should be stored in -20, or they should be amplified immediately for library preparation. The fragmented structure of this cell-free DNA molecules are also oblige the genetics laboratory to adapt their amplification protocols. In fact, some small modifications or also for the amplification protocols has been done and should be validated. And after our validation study for the concordance analysis, we applied our method for 68 spent culture media cases coming from the blastocyst level embryo, just to understand if there was a difference between day 4, day 5, or day 4 to day 6 amplification rate and informativity rate. These are very not a big number, are small numbers, but we saw a very similar amplification rate and informativity rate between day 4, day 5, and day 4, day 6 groups. This means that the 24 from the 24 hour and more, we can get quite satisfying results for amplification and informativity. Another factor we just checked if there was a difference between the medium brand. We tested the 2 different medium brand, but the one is about 55 samples and another was only for 13 samples. So the numbers were not equal, but even using the different spent culture media samples, we observed a very compatible amplification and information rate. And another parameter we checked for the performance of non-invasive PGT was the embryo morphology. Of course, we classified the embryo samples like grade A, AB, or BB, or grade C. Again, here we also observed the cell-free DNA analysis performance was quite similar between each groups, and especially for grade C group, there was only 7 embryos, but they gave 100% of amplification and informativity rate. So it is very small number. So statistically, we cannot say, but we expect that, of course, if the morphology is not very good, this may also be an indication of the presence of apoptotic cells. So that's why maybe we get a very high rate of amplification and informativity. So the main question is if may cell-free DNA analysis culture media replace trophectoderm biopsy. Of course, there are some studies, promising studies, is showing that higher sensitivity and specificity rate when, for instance, in this study, spent culture media and trophectoderm biopsy samples results as being confirmed, compared with the whole embryo results. And the non-invasive PGT-A approach was giving a higher sensitivity and specificity than trophectoderm biopsy samples. But of course, this was only 50 samples, and it was a frozen thought cycle. So a little bit, this can be criticized, but like this is not a real scenario for the routine IVF practice. And another sample, another study is only with 10 samples. Again, non-invasive PGT-A sensitivity and specificity were higher in the non-invasive PGT-A group. And another study from 2019 by Carmen Rubia and his colleagues showed that especially the clinical pregnancy and ongoing pregnancy implantation rate was higher in the sample group where spent culture media and the trophectoderm samples were both haploid, and the less the performant clinical data has been observed if the spent culture media showed an aneuploid and trophectoderm sample was haploid. So this is also very small number, but can be a good indicator for the performance of clinical application of non-invasive PGT. So, as a modeling of a comprehensive non-invasive PGT approach should be an ideal approach. And if we look at today's application, the non-invasive PGT-A can be an alternative for embryo prioritization. According to the haploid profile, we can give a prioritization score for each embryo. So in that way, where the embryo biopsy for the invasive method is not possible, we can give an opportunity for the ranking the embryos according to non-invasive PGT-A results. And of course, there are some other approaches like embryo prediction by morphokinetic analysis. And there are also several studies has been already published via very good promising results. And these studies are showing very near prediction values for the aneuploid detection. But probably the most suitable approach should be the combination of these two different applications, like the genetic testing by non-invasive PGT-A and embryo morphokinetics, and together with some other parameters like previous clinical outcome or clinical indication of each patient. All these data can be combined with an artificial intelligence algorithm for the embryo prioritization to select best competent embryo. As conclusion, all recent studies now demonstrating us the potential use of the cell-free DNA as an alternative to invasive PGT methods. And of course, to be able to apply in a correct way, all current IVF laboratory protocols need some modifications and to adapt for their adaptation to non-invasive PGT applications. Like each genetic test protocol, also the non-invasive PGT applications needs validation tests, and the standardization of some methods are very important, especially because today's standard invasive PGT methods are mostly designed for the amplification of targeted genomic DNA molecules from single or a few cells. But cell-free DNA molecules and their structure are totally different. And the non-invasive PGT can be used today as a scoring system for the embryo prioritization to select the embryos with higher chance of haploidy. But of course, we need some randomized clinical trial results, which are some studies are ongoing for the robust and safe, effective use of non-invasive PGT-A in clinical practice, and a comprehensive non-invasive screening techniques combining genetic and morphokinetic and clinical data processed by an artificial intelligence algorithm will provide us more robust data on the selection of most suitable embryo for transfer. I would like to thank my team in Eurofins Laboratory and also for collaborating centers like Drs. Veronica Bianchi, Professor Emiliano Greco, and for all other contributing laboratories to this study. And thank you for your attention.
Thank you, Anil. Thank you, Anil, for this very well and detailed presentation. I think, as you mentioned, it will be like for the moment, non-invasive PGT-A could be used as a scoring for embryos, or whenever like the trophectoderm biopsy is not feasible. We have a lot of questions, so stay with us for the Q&A because we have a lot of questions to ask for you. And in the meantime, just relax and let us introduce our next speaker, which will be talking about the practical experiences on PGT, tubing, and genetic sampling of the blastocoel. Dr. Evrim Unsal is our next speaker. She is an embryologist since 1997. She received her PhD in biotechnology, specialized in reproductive genetics. She became an associate professor in 2016, lecturing in medical genetics. As a molecular biologist, she is director of Microgen Genetic Diagnostic Lab, focusing on PGT. Currently, or recently, she was an associate research scientist at Yale University Fertility Clinic from August 2023 to August 2024. And nowadays, she continues her position leading the molecular genetics department at Microgen Genetic Diagnostic Center in Ankara, Turkey. Dr. Unsal, thank you so much for joining us, and please go ahead with your presentation.
Thank you, Berthe. It's my pleasure to be here. And actually, first of all, I'd like to thank Dr. Jill Palmer for inviting me to participate in this webinar. And I'm glad to have the opportunity to share my work here. And I appreciate everyone who have contributed to organization this event. And my disclaimer for today is like that. And first of all, I wanted to share with you the PGT Good Practice Guideline prepared by the Turkish Society of Reproductive Medicine Genetics Special Interest Group, which I was coordinating this group, and a consortium of 26 experts in the field of PGT. And the summary report focusing on just PGT-M session has been published in RBM Online, and all the attendees can also access the full version on the TSRM website. During my presentation, I will explain key points about blastocoel fluid and its role in PGT-A. And I'll start by defining what blastocoel fluid is and how it's collected in PGT-A procedures. And I'll also highlight the advantages and limitations of using blastocoel fluid for genetic testing. And after that, I'll discuss why PGT-A is controversial and how we can improve the efficacy of this test. Since I am the last speaker, some of the content may be repeated, but I'll present it as I have prepared it for the integrity of the presentation. The presence of DNA in blastocoel fluid was first reported in 2013 and opening a new era while discussions about spent culture media were ongoing. And the collection of BF-DNA is not completely non-invasive. And the key question remains if this DNA accurately reflects the genetic status of embryos. At that time, discussions were ongoing about the accuracy of the test and the potential detrimental effects of blastomere or trophectoderm biopsy on the embryo. And as you see over here, the reported concordance between T biopsies and the spent culture media showed significant variability, ranging from 33% to 90% almost. So similar variability in reported concordance was also observed in BF-DNA, and these discrepancies may be related to differences in different culture protocols and DNA testing methods, potential external or maternal contaminations. While it remains a promising approach for embryonic genetic testing, it's still under investigation and cannot be offered as a diagnostic test until the origin of the DNA is clarified and concordance results are reliable as with at least as with T cells. Today, it's been adopted by some laboratories as a tool for prioritization and defining the competent embryos for the transfer. In a study published by Dr. Magli and her colleagues, blastocoel fluid biopsies were performed and 30% of these samples couldn't amplify successfully. 94% concordance rate was observed between aneuploid status of T cells and BF. However, of these total concordance, 66% was reported as full concordant and rest is partial concordant. And in this study, overall amplification success rate was 21% across all samples. However, 55% of these euploid embryos failed to amplify DNA, while 19% of the aneuploid embryos revealed amplification failure. According to researchers, these findings can be used for selecting the most viable embryo for transfer, and they speculate that priority would be given to T euploid blastocysts having BF with amplification failure. As you know, the search for markers such as time-lapse prediction, metabolomic testing, morphokinetics to obtain a competent embryo has been going on. And the embryos with non-amplified results would be selected for further TE biopsy, allowing only pre-screened embryos to undergo PGT-A. However, considering that even the TE biopsy process itself remains controversial, and when the factors such as cost effectiveness and the potential risks to embryo are considered, I believe this approach may not easily become a part of routine clinical practice. Technically, since spent culture media is a non-invasive method, the semi-invasive blastocoel fluid biopsy has actually only been the focus of limited study groups. And although many studies have looked into SEM samples, no established method for routine clinical use has been introduced yet. And the study I'm referring to actually focused on comparing the effectiveness of SEM and the BF samples by looking at the ploidy concordance of both techniques. And I won't dig into details to save time, but briefly, they found that SEM samples showed more than 5 times higher pre-VGA DNA content compared to BF samples and higher concordance rate with the blastocyst and the inner cell mass. And according to me, since acceptable consistency rates have been achieved, SEM technology isn't quite ready for the routine clinical application. In this context, many research groups suggest that SEM technology could be used as a way to prioritize embryos with higher pregnancy potential. However, because it's difficult to standardize sample source across IVF labs, these methods are not yet ready to become routine in genetic tests. Building on challenges and limitations we have discussed, it's important to note that the PGT consortium has reported that these techniques are still in the validation phase, and this means that further optimization is needed before they can be widely adopted. And when we look at the nature of blastocoel fluid, it's like a collection of various apoptotic cell products like proteins, mtDNA, mRNA, and DNA. And these DNA fragments ranging from 160 to 220 base pair in length are very similar in nature to typical apoptotic fragments circulating in the blood. So BF DNA often originates from necrotic or apoptotic cells, and this can affect both quantity and quality of DNA. And this leads to significant variation in amplification rates and concordance with TE biopsies. Differences in technical expertise in performing blastosynthesis is also an issue for the inconsistency. And for these reasons, despite the potential clinical benefits of blastosynthesis, its use is still under investigation and not fully reliable for routine testing in current state. Technical limitations and heterogeneity in technical expertise with blastosynthesis contribute to difficulties in assessing its validity. In current methods, all the fluid from blastocoel cavity is aspirated using an ICSI pipette. However, transferring this fluid into the PCR tube without losing any amount is technically challenging. Some practitioners use a very small droplet on the Petri dish during the biopsy, then try to transfer the fluid into the droplet before moving it to the PCR tube. This method can still result in DNA loss during the transfer from droplet to tube. When trying to transfer the fluid directly from the ICSI pipette to the PCR tube, there is a risk of breaking the glass pipettes, which can cause issues that might inhibit PCR reaction. And in order to avoid stiffness of the fluid, a preloaded microneedle with media can be used, and this volume will also provide extra pressure while releasing the fluid into the tube. An additional manipulation and cost burdens, such as needle replacement for each embryo biopsy, limits the methods as well. And actually, the tubing protocol is always an issue in PGT-A applications. And to address this, Leo and his colleagues introduced a tool you can see over here, and that allows direct transfer of the biopsy trophectoderm cells into the PCR tube under an inverted microscope during manipulation. And to avoid sample loss during BF transfer, such a tool can also be used to increase DNA yield and improve the accuracy of PGT-A testing. With this approach, a more precise concordance study can be performed for BF DNA as well. Despite the ongoing debate about PGT-A, as you can see from our NGS-based PGT-A embryo referrals, demand has been increasing every year. And this graph shows data from the first quarter of 2024. And of course, like a techniques, PGT-A has some advantages and also disadvantages. Thanks to possibility of selecting euploid embryos before transfer, PGT-A provides a higher possibility of successful pregnancy and reduction in spontaneous miscarriages. Among many different reasons, PGT-A is still controversial, and in particular, nowadays false positive results has become a significant point of debate among IVF practitioners. One of the key issues in these controversies is the retesting of embryos that were initially reported as chaotic. Interestingly, some of these embryos end up showing euploid results after rebiopsy. For example, in this study, 4% of 3,000 embryos tested via PGT-A were initially identified as chaotic. When 54 of these embryos were retested with a new biopsy sample, 4 remained chaotic, but 9 were found to be euploid. And similarly, an abstract from Turkey showed that samples reported as chaotic or inconclusive by one laboratory yielded different results when tested at the other laboratories. To assess the controversial outcomes of PGT-A, we need to identify the potential sources of these problems. These issues should be assessed by both IVF and genetic perspectives. Unfortunately, since there are only a limited number of reproductive genetic laboratories, these problems are often raised by IVF practitioners in the literature, and the assessments may lack the viewpoint of genetic labs. As a former embryologist with the experience from both perspectives, I'll briefly summarize the situation in the next few slides. First, these evaluations must be evidence-based, and each laboratory should use KPIs and benchmarks. With this approach, my colleague Dr. Bashar from Yale University prepared a review on IVF KPIs, and I contributed to the section of PGT KPIs. The first step always is a physical examination of PCR tubes, followed by whole genome amplification. And the samples are then sequenced using NGS, where MAPD values, productive read counts, and quality scores gives us quantitative way to assess sample quality. When the PCR tubes with the biopsy samples first arrive at the genetic laboratory, the first step is a physical examination. when performing cell tubing on 2 microliters of PBS, adding extra volume can affect the PCR reaction and increase the chances of false positive results. Similarly, during Histolget 2022 PGDIS Congress in Berlin, Dr. Richard Scott mentioned that in their lab, different embryologists showed variations in their tubing techniques. He pointed out that when tubes with an increased volume, like 3 microliter, were used for NGS, the MAPD values were affected. Since these types of samples are valuable material, genetic laboratories conditionally accept these inappropriate samples after physical examination. The second stage of quality control is the assessment of amplification success after whole genome amplification. This is done by either spectrophotometric measurement or gel imaging, depending on the platform used. And here you see a batch of 96 samples, almost all of which were successfully amplified. However, tubes marked in red indicate amplification failure here, and while those marked in blue indicate weak amplification. MAPD is one of the most important quality control metric used to assess the noise level in next generation sequencing data, and it evaluates the variation between the coverage of the probes across the genome. And lower MAPD score indicates less noise and better quality data, while higher MAPD score suggests more noise and potentially unreliable data, and which can affect the accuracy of PGT-A tests. Generally, a MAPD score should be below 0.3 is acceptable for high quality data in PGT-A analysis. Here you can see cytoviews of the cells with different amplification level. And the first image is from a well-amplified sample, and MAPD value is 0.2, and this is a very important KPI to evaluate sample quality. Weakly amplified samples mostly reveals NGS biases. In the second view, gender information is missing, and due to low quality, X and Y chromosome log ratios cannot be accurately reported. The last one is from a sample with amplification failure, and last 4 samples are inconclusive due to insufficient MAPD value and cannot properly interpreted. When we see these bias aneuploidies, doing a second analysis with a new biopsy might actually show euploid results or different results. To keep short, sample quality is the most important factor for getting accurate results with the PGT-A technique. And the read count is another crucial KPI. For PGT-A, a read count of over 70,000 is needed to ensure accurate results. As you can see here, samples with low read counts can be reported as inconclusive due to poor data quality. And here there are these highlighted 3 embryos from the same patient were discarded due to ineffective tubing. However, highly reproducible results were achieved in the remaining patients from the same NGS run. It should be acknowledged that the rates of mosaicism, segmental aneuploidies, and inconclusive embryos in PGT-A testing are operator-dependent procedures. Reference genetic laboratories that provide testing services for many IVF clinics, they run 96 or 192 embryos simultaneously. So we can clearly identify differences among centers and even between embryologists within the same IVF clinic. And the KPIs I mentioned play a crucial role in providing feedback to IVF laboratories. In fact, these evidence-based metrics have recently been reported by leading groups conducting significant research in this field, and this study The quality assessment of biopsy and tubing techniques by embryologists working in the same labs were evaluated at the NGS stage, and attention was drawn to the deviation that contribute to NGS biases. Since we have been giving service to IVF laboratories in 42 different countries, we have been experiencing such a different bias and no result ratios among the centers. And these technical errors can be arise during biopsy or tubing stages. And we always give feedback when we see unexpected fluctuations. And as you see in gel electrophoresis result over here, first 7 samples didn't amplify properly. And when we investigate the root cause, we found out that calcium-magnesium-free media was used for biopsy, which inhibits PCR reaction during VGA. Additionally, optimal washing procedure should be performed in order to avoid such a inhibiting contamination of these inhibiting factors, and vigorous washing should be performed by the embryologist before tubing. The other faulty biopsy approach is to remove leftovers of cell debris instead of TE cells for the intention of minimal invasiveness. These debris are not reflecting the embryo reality and reveal false positive results. Tubing of the biopsy cells has a great impact on PGT-A results, and from our experience of working with IVF laboratories that perform different tubing approaches, standardization of tubing method is crucial for robust genetic test results. Inappropriate tubing always reveals inconsistent results. And actually, we shouldn't transfer biopsy cell batch without washing vigorously. We do not perform tubing at eye level, and we should always follow the releasing of the cell into the tube under the stereo microscope. And we shouldn't perform tubing while any of our colleague is making vitrification in the same cabinet, and keep away our tubes from the liquid biopsy. And we should remove cumulus cells efficiently, especially in PGTM cases, and take cautions to avoid external contamination, and transfer biopsy cells with a minimal amount of media, which is very important one. And we should change the tips for each embryos to avoid any contamination as well. And there's a limited studies focusing on inappropriate tubing effect. And for this purpose, we design a multi-center comparison study to evaluate impacts of different methodologies on genetic outcomes. For this purpose, we have been comparing conventional tubing method, which is cells are transferred to PCR tubes containing 2 microliter molecular grade PBS, and the dry tubing method, cells are transferred directly to the bottom of the empty tube, and the droplet method, cells are transferred in the microdroplet made at the bottom of the tube. And the colleagues from Yale University, we conducted embryo biopsy for donor embryos for research aim. And according to preliminary results, with the 48 sample, we obtained successful amplification in all these samples. You may see the initial samples and the gel view of the whole genome amplification products. Among all methods, no major inconcordance was observed, apart from minor differences in mosaicism results, which will be evaluated once full data set is ready. Because we are gonna perform a comparison study for 400 samples, trophectoderm biopsy samples, and we will compare these different types of tubing methodologies. And only one sample we have detected a segmental duplication with a single method, which is considered as a technical artifact. And once the project is finalized, we will publish the efficacy of each methodologies. I'd like to thank my teammates and the owner of the Microgen, Professor Dr. Baltacı, and thank you for your attention. And if you have any questions or would like to discuss this topic further, feel free to reach out to me and I'd be happy to connect.
Thank you very much, Dr. Unsal, Evrim. That was a really thorough talk about how important it is for us to get down to the basics and correct the technologies that we use with key performance indicators and so on. It's very important to understand the technology as well as the biology in these very demanding tests that we're trying to do on preimplantation embryos for their predictive potential. I would like to ask you a little question. You mentioned at some point the volume of the blastocoel fluid that we are able to extract and analyze. What was it in the range of nanoliters? I seem to think.
Actually, I think Professor Biricik mentioned about it.
Yes.
And I think they have such a experiment. Maybe he may give a response. However, the major point of my focus is to obtain whole fluid and collect them into the tube. And there are several problems like thickness and also transferring from the transferring directly into the tube. And the tool I showed you can be helpful. Giving the samples directly into the PCR tube under the inverted microscope. This is very crucial according to me.
Yes, because we're talking about such tiny volumes of liquid, and any loss is obviously going to be very detrimental to the potential of a genetic analysis. Now, I think it's time for us to open up the question and answer session. So all the speakers, if they'd like to turn on their cameras again, and also my co-hosts as well, so that we can ask some questions. I'm going to pick a question from the many questions that we got sent through the question and answer site. I'm going to start with some of the questions that were directed to Dr. Nakhuda, who was the first speaker, and from somebody called Bronte Stone. They don't identify where they're from. They want to ask Dr. Nakhuda, in the recent NICEST report, it's presumed that only morphologically normal blastocysts were transferred while blinded to ploidy. And against this partially blinded study, did outcomes reveal any interaction of ploidy in association between morphology and outcome?
Thank you for the question. I'll try to answer. So, we just use a routine embryology criteria for selecting embryos for freezing and transfer. In order to meet criteria to freeze, the embryo had to have an expansion grade of 4 and a grade of at least a B in either the ICM or the TE, and it could have a C or above for the other grade. So in the cohort of embryos that were studied, there weren't any real differences that we can discern in the grading per se, because it's fairly widely dispersed. But the association that was relevant was the day of the freeze. So day 5, 6 versus 7. So in terms of the informative data, there was basically every single day 7 had at least passed QC, and both in the cohort of the, so we studied a total of 825 embryos, right? Only 120 were transferred. And there were 0 day 7s that failed QC. There was only 1 day 7 in the cohort that was transferred and that did not implant. But if again, we look at the day 5s versus day 6s versus day 7s, and this is consistent, I think, with every study, and Dr. Biricik alluded to this, the longer the time in study, the more informative the data. So I don't know if that directly answers the question, but you know, the results seem to be more accurate the further the embryos are cultured.
Thank you very much. Catherine, would you like to answer or ask
Sure,
her?
we'll ask another question. I think this can probably be for any of the speakers. Is it necessary to perform zona drilling on the embryo, and do we need to wait until the blastocyst is fully expanded or hatched?
Who would like to answer that question?
Maybe I can give my answer.
Okay.
Yeah, okay. Yeah, basically, okay, zona drilling for assisted hatching may help to release of the blastocoel, also fluid in the span culture media, but it is not obligatory. And in our study, we get really high performance results even with
I think
embryo culture without any cyst. Yeah.
I
Can you hear me?
Well, no, I'm not sure if your connection is unstable. Was it mine or was it Anil's?
Can you hear my answer?
It wasn't very clear, Anil, actually. But I'm not sure if it was my connection or not. I think maybe it was yours. Catherine?
I can. Yeah, basically, I see my connection is quite okay, but I don't know if the other can hear me.
Can anybody else hear Anil?
It was very choppy.
Yeah, okay.
Oh, sorry.
It's okay. I don't know. Beta, would you like to try again, Anil, to answer?
Yes.
Yeah. Okay.
Let me try again. Can you hear me well now?
Yes, we can, yes.
Okay. Maybe, I don't know, maybe probably, yeah, my connection. I'm sorry for that. Yeah, actually, I was saying that, okay, zona drilling is not obligatory. So but of course, if it has been done, then we know that the release of the cell-free DNA in the from blastocoel fluid in the span culture media can increase the concentration of cell-free DNA molecule. And usually in our study, we also tested several embryos without zona drilling, and we get very good results. The critical point is the culture time. So if the culture time is at least 48 hours, we get really good amplification and conclusive results rates. So this will be my comment. I hope you could hear well.
Okay, no, that was good.
Thank you, Anil. That was crystal clear. Thank you so much for your answer. We have many questions from Helen in the chat. So I will direct the next question to you, Dr. Unsal. And Helen is saying to us, dear colleagues, thank you so much for the talks, giving the critical role of repetitive DNA in advancing non-invasive genetic sampling. She would like to know, how do repetitive DNA sequences influence the accuracy and reliability of non-invasive pre-implantation genetic testing? Maybe could you comment on this, Evrim, please?
Actually, while she's saying to repetitive DNA, actually, what is the
She's just wondering how the repetitive DNA elements contribute to the clinical outcome of IVF treatment in general, and if it has an influence while doing non-invasive preimplantation genetic testing.
If she asking about satellite DNA and actually maybe these repetitive DNAs actually selecting embryonic cell-free fractions may be an issue. And but there, I don't really have any idea. These repetitive DNAs also helpful to evaluate these embryonic cell-free DNA fractions. I don't know if Dr. Arıcı or Biricik or the other speaker may have information about it.
Yes, Anil, would you like to have a go? But I'm not sure that the question is very clear.
Yeah, I'm not sure about the
Yes.
what about the repetitive DNA. I don't know if it is a
Yes.
question regarding the
Biology or the technicality of actually doing the analysis, because we use repetitive DNA when we do STR analysis for
exactly.
Yeah, actually, for PGTM, of course, we use it in our routine,
Yeah.
but for non-invasive approach, some researchers also focus on to identify cell-free DNA in the bioinformatics stages by using SNP profiling or not SDR, actually, SNP profiling. This is a unique approach, but if the colleague asking this question has an idea, maybe she or he can give details. And the repetitive DNA usage is not a well-known issue for non-invasive PGT, but also PGTM mostly.
I can give just my contribution. Sorry.
Of course.
Yeah, basically in our studies we use some, if they their question about the short tandem repeat markers, if so, then yes, we also use these markers for contamination detection, and also some trials for PGT4 monogenic disorders, like linkage-based studies. Also, these can be used because they are very small. If they're very small amplicon fragment sizes applied, then you can also detect these small short tandem repeat markers in cell-free DNA analysis. I don't know if the question is asking this feature of use short tandem repeat markers. Yes, there is a possibility to use these markers both for contamination detection and for like fingerprinting. I can say that to understand if the content of cell-free DNA is coming from the parents of the embryo.
Mm-hmm.
Yeah, I was just going to say I'm a bit out of my depth of the question, but that's how I was going to interpret it and what we were thinking about in order to determine if there's contamination or not. So that's an interesting question.
Yeah, I kind of associated with this approach, this question. I'm going to ask everybody from Dimitra Christopoulou. Hi, Dimitra in Athens. She thanks you all for your presentations. And she's asking, would you see in the future the application of a technique that could simultaneously test for CNVs, SNP haplotyping for better analysis between meiotic aneuploidies and mosaics, and whether on that basis, in fact, we could also extend it to do non-invasive preimplantation genetic testing for monogenic diseases? So I think probably if we'd like to go in the order of the speakers, perhaps for this one.
Yes, I again, out of my depth, I'm just a clinician, but this is something we've talked to the laboratory about. If we can use SNPs and haplotyping, hopefully it can help at least rule out maternal contamination. Essentially use it as it could theoretically use it as a reflex test. If we saw 46XX, we could determine how much maternal contamination is, and then do further analysis from there. And just to say, there have been case reports of using cell-free DNA from the spent culture media for PGTM.
Yeah.
It's also been used for structural rearrangements. And again, even in our case series, we detected the aneuploidy, not exactly the right structural aneuploidy, but the correct chromosome. So I do think the potential is there.
Okay, Anil?
Maybe also,
Oh, sorry,
Yeah.
go ahead.
actually,
Please, Dr. Unsal.
instead of aneuploidy screening, making a targeted analysis for monogenic diseases can be more reliable approach for with this sample source. However, we mostly focus on PGT-A, but it can be also implemented using these samples for the monogenic disease, PGT. Since we make direct analysis for target mutations and also STR or SMP profiling, and it can be more accurate according to me. And higher responsibility needs as well.
Sure. Anil?
Yes, basically, I agree with Dr. Unsal. First of all, the criteria for the QC criteria and the validation results of such kind of approach should fulfill the current standards recommendations for, especially for single gene disorders, because we are talking about the diagnostic approach in this point. So it is not a aneuploidy screening and the prioritization. So that's why technically the protocols should be very well validated. And if we are from technical side of view, we are talking about very small fragments. And currently, the best recommendations for single gene disorders is like using the multiple displacement amplification for a good coverage of the genes and also to avoid, to reduce the allelic dropout rate and to avoid misdiagnosis. And if we during my presentation in one of my slides, I just tried to underline this: the amplification protocols should be very well optimized using if the very deep sequencing NGS protocols, if they can be used, but the small fragment sizes for the library preparation, the preparation, and a very good coverage of the targeted genes together with flanking the markers like SNP markers should be covered very well. So I think, yes, the ideal will be one unique protocol, non-invasive protocol with covering copy number analysis, single gene disorders, the single nucleotide polymorphisms, and also single nucleotide variations to detect all in one. But the protocols should be very well adapted and validated for this approach.
And we must never forget the aim of the test.
Absolutely. Most importantly.
The more we look for, the less normal embryos we'll be able to find. So, I mean, this is also very important, I would say.
Absolutely.
And associated to that, we had a comment from somebody called Murat Sedtinkaya, who says it's not directly a question, but rather a comment on Dr. Nakhuda's talk. And he agrees that niPGT-A is not a binary test in terms of the result, but it gives a spectrum and the prioritization of the available embryos. And so the interpretation must also be very carefully explained to the couples, and follow-up testing, at least in the form of NIPT, should be recommended. And I think, in fact, Dr. Nakhuda, you did say that in your talk.
Yes, I completely agree. I think, there may be certain results where we have extreme confidence that the determinism of that result, if it's a whole chromosome aneuploidy, it's well above the thresholds, as we even demonstrated in our little study. I think we can say that and explain that to patients. However, I think also as a field, we have to stop pretending that we have a perfect answer on each of these analytic profiles, right? We just, okay, look, we don't know what this means. It's we may have to set other thresholds for failed QC. If that's the difference between somebody potentially having an embryo and discarding one that we're not, certain about, I'm certainly don't want to be the arbiter of that decision, right? I want to let the patient have that decision and be honest about what the results are. And so I think that translates to all the testing that we do, including trophectoderm biopsy and everything that comes in the future.
Sure.
Great. So maybe we move to a more back to the lab a little bit, and a practical question for looking at the contamination, especially the maternal contamination. What steps are being made to control for minimizing that contamination? And again, in any of your labs, so maybe want to go through the speakers again.
Can we follow off the order of the speakers, or I can give my answer?
Yeah, go ahead, Dr.
Go ahead.
Right. Well, I can start, but again, not an embryologist, so I'm out of my depth. We tried to keep things aligned with our embryology lab protocols as much as possible. So, the best practices there, working in the hoods, gloves, etc., masks, that type of thing. I would say we have as many men working in the laboratory as we do women. So unless the conclusion from our study is that the men are better at getting cleaner samples than the women because there's more contamination from XX, I'm a little bit reluctant to say that, especially to this crowd.
Yeah, actually, I will talk about our experience until now. During our validation study, we analyzed. Now today we have more than 500 samples during this pilot study we applied, and frankly speaking, we didn't get any contamination from the blank media sample. So this means that the IVF lab, if they use the correct protections and the correct protocol, we are very comfortable about the external contamination. But the problem is the maternal contamination, and the like 5% of false negative samples we obtained, they were all having the female profiles during our concordance analysis study. So this means that the maternal cumulus cells contamination is a big issue. So today, the only way is to mechanically to remove the cumulus cells and to do the sequential medium washes before to put the embryo last droplet for the final development stage, which it will release his cell-free DNA. So despite this, we can reduce, of course, the maternal contamination, but other than we have to also find a way to detect this contamination, at least if when we detect it, then we can give at least not conclusive results. So today, this could be the main solutions to avoid contamination.
Actually, in the IVF laboratories, for PGT-A, we were using additional vigorous cumulus cell removing. What we were doing while in the first hyaluronidase denudation step, we were keeping embryos incubator for one more hour. So 1 hour more, sorry. So it allows cumulus cells to release, and making additional mechanical denudation after these extra incubation time with the extra incubation times allow us to remove almost all the cumulus cells. This kind of extra stage can be also implemented, and the second or third day culture media changing will be also helpful to avoid maternal contamination, to remove cumulus cells actually.
Wonderful. Thank you. Well, I think it's time to wrap up. So thank you. I wanted to definitely thank all of the speakers and the hosts who took part in this webinar. Of course, my co-host, Dr. Berta Yunus, and Professor Jan Traeger-Synadinos. The producers, the presenters, Dr. Gary Nakhuda, Dr. Anil Biricik, and Dr. Evrim Unsal. And of course, a very big thanks to Thermo Fisher Scientific for sponsoring this very enlightening webinar. We've really enjoyed the last couple hours.
Yeah, thank you, Catherine. And also, we mustn't forget the team from I3 and the organizing committee of this initiative, who include Jack Cohen and Peter Nagy and Marianne Sevetes. Sorry, Marianne. Thomas Elliott, Fran Fali, Violetta Sora, and Giles Palmer, of course.
Thank you so much from my side as well. And in October 1st, it's really soon, you will have another webinar from I3. So join us soon in the next I3 session. It will be about vitrification unlocked, advanced vitrification for gametes and embryos. So save the date. It will be on the 1st of October. Don't miss it, and see you soon. Thank you so much for joining, and hopefully we will see each other in the future webinar. Bye.
Bye-bye for everybody. Thank you for
Thank
joining.
you.
Bye.
Thank
Bye.
you.
Thank you.